Hey all, good morning. Is this working? Can everyone hear me? Hey, so it's a tremendous honor to be here in Varna and be able to tell you about some of the work that we've been doing in my group and, broadly reflect a little bit on what's happening in the field as a whole. I'm going to talk about 3D genomics, which is basically the use of genomic methods to interrogate how the human genome folds in 3D. And I'm going to really focus on there's a lot of different things that are going on, but I'm really going to focus on two problems and the application of 3D genomics to solve two very specific problems. One of them is a problem that is actually about how the genome folds in 3D. That's the problem of how to map loops, and that's going to be the topic of my first talk. And then the topic of my second talk is going to be a demonstration of the ability of methods derived from 3D genomics to solve problems that superficially have nothing to do with nuclear architecture, have nothing to do with how the genome is folded in 3D, and specifically solving the problem of how do you, in a fast and cost-effective fashion, assemble new genomes, do roughly what the Human Genome Project did, but much faster and cheaper. Okay, so with that sort of table of contents, you should know which of either of the talks you're interested in. Of course, you can walk out after the first or into the second or whatever, depending on what your preferences are. So anyways, I'm going to start by telling you about the use of 3D genomics to probe the problem of mapping loops. And I'm going to start with some really, really basic facts just to make sure that everyone's on the same page. And sort of in the spirit of this is a bit of a pedagogical session. So the problem of how genomes fold is pretty simple to describe. Basically, the human genome is about this big, and yet it folds up to fit inside the nucleus of the cell, which is only a few microns wide. And so the question is, how does it manage to do that? That's the problem of 3D genomics. And this problem is not purely of interest from the standpoint of physics. It's been of great interest to folks interested in epigenetics, folks interested in the determination of cell state, because it's well known that you have roughly the same genome in all of your cells, but your cells do very, very different functions. That's because different genes are on in different cells. And well, why is that? That's a very basic question. In the late 70s and early 80s, it was discovered that there were these strange DNA elements that could live far away from a gene, but yet seem to be able to turn it on and off. This was not initially believed, but eventually people figured out that this was real, and so they named these things enhancer elements. And so that immediately raised the question of, well, how in the world is it that an enhancer element can regulate a gene, even if that enhancer element may live 100,000 base pairs away, or 1 million base pairs away, or more? And the hypothesis that emerged pretty much immediately was that the way this worked was that enhancers actually formed loops to the target promoter in 3D. So even though the enhancer was far away in one dimension, it was actually adjacent in 3D, and so that's sort of how this spooky action at a distance managed to work. And so for that reason, the problem of nuclear architecture was not purely a problem of interest to physicists, but was also really a problem that was of great urgency for the understanding of cell biology for the last 40 years or so. Anyway, in general, if you're looking, trying to figure out the structure of a system, a great way to do that is using microscopes if the system is small, or telescopes if the system is far. It's really been the case for hundreds of years, and I'm sure it's going to continue pretty much indefinitely. Like, I don't know, what would happen if, like, a time traveler landed on Earth in 500 years from now and said something like, "Take me to your leader." I'm sure the people would be very confused and say, we don't even know what the word leader means. We're like in a anarcho-syndicalist commune. That doesn't make any sense to us. But I'm completely certain that 500 years from now, you said, "Take me to your microscope," people will be like, "Oh yeah, we know what you're talking about." And they'd show them the latest microscope and all the improvements in resolution that they've managed, because it's a very good technology. Nevertheless, a occasionally it's not actually the only way to do this sort of thing, and so I wanted to highlight for you this sort of concept of contact mapping, which is sort of the notion that you can explore the structure of a system by exploring proximity relationships within the system. And as an example, I like to use this individual. He's very recognizable in many places. He lives in the fictional town of Springfield. And if you are interested in studying the structure of the fictional town of Springfield, then you could kind of use this sort of contact mapping approach. And how would that go? Well, what you would do is you would take sort of pictures, sort of surveil Springfield, and try to find cases where the individuals are sort of connected with one another in some way. So here he is connected with a donut. It's actually not so helpful from a sociological standpoint. This is more interesting. Here is this individual and that individual. I don't know what they're up to, but seem to be up to something. More individuals, more individuals. Eventually I have a lot of data. It no longer fits on a slide, and so you need to figure out what you're going to do with it. And a simple and kind of moronic thing that you can do with it is you can just put it in like a matrix, a table, which shows, for instance, in the data that I just was showing on the last slide, this is just a reflection of that data. See that this individual and that individual have an enhanced frequency of proximity with one another. So they are, in fact, statistically speaking, up to something. And whatever it is they're up to appears to have nucleated a whole family of individuals with enhanced contact frequency. Anyway, this is kind of a very silly thing and really a waste of your time in a lot of ways, except for the fact that you can actually do this sort of thing with the human genome. This was demonstrated in a classic experiment by Catherine Cullen and others in Science in 1993. I'm going to abstract away from biology a little bit, just not to get you bogged down in the details. They hypothesized that these two elements, which they drew as a Pac-Man and as a power-up, formed loops. That was their hypothesis, and they noticed that these two elements were really, really close to restriction sites of the restriction enzyme PST1. And so they had the following idea. They said, well, why don't we use this restriction enzyme and cut at the PST1 site, and then we'll introduce some T4 DNA ligase into the tube. And T4 DNA ligase, what it does is when it sees free DNA ends, it fuses them together and produces a ligation junction. It's interesting that I'm able to move the mouse somehow. That's spooky action at a distance, I told you. Who is doing this? Wow. Anyway, spirits of the genome. Can you like move it to like here? I'll just use my finger. Okay. I'm sorry, that wasn't. This wasn't reversed. So they said, look, we're going to introduce the T4 DNA ligase. What T4 DNA ligase does, when it sees two free DNA ends, it fuses them together. It ligates them to form a ligation junction. And their idea was, if you first restrict and then ligate, then if, in fact, these things are in the looped configuration, then the two PST1 sites will be drawn very close to one another. They will ligate very, very frequently, and you'll have a really high frequency of this ligation junction fusing the two DNA ends. They said, by contrast, if Pac-Man is not in fact biting down in the power-up, and they're not actually forming a loop, then you would rarely get these ligation junctions. And they would measure the frequency of these ligation junctions in various ways. They'd sequence the junctions, they'd do gel electrophoresis on the junctions, and all kinds of different measurements for doing this. And this was actually a really, really important experiment because it demonstrated. Okay, well, we did an experiment. It's this thick somehow. Anyway, well, it demonstrated that, please just ignore that. I don't know why I'm doing it, but I am. So it demonstrated that you can do contact mapping on the human genome. And so the question is, like, why didn't people give the talks I'm about to give now, 25 years ago, and it's because there were really two fundamental challenges. And the two fundamental challenges were, first, that without a reference genome, these kinds of experiments are very, very hard to design. And in 1993, there was no human reference genome. And the second is that without cheap DNA sequencing, these results are very hard to quantify. Because you'd have to do things like PCR against every locus that you're interested in, and this was sort of a pain in the neck and very time-consuming. Sorry, you guys will have to put up with the nuisance of that thing. We have figured out that, in fact, I am responsible for it, but I don't know how to stop it. So the point, today, I mean, it's possible to do these kinds of experiments much, much more efficiently. And the way that you do them, I mean, this sort of experiments have been refined many, many times. And there's the chromosome conformation capture, 3C, and 4C, 5C. I was in graduate school. I came up with this method, which together with others we implemented, called Hi-C, which is just basically a fancy way to do nuclear ligation assay and read off the frequency of all the ligation junctions at once. The basic idea is if I have a pair of loci and they're in close contact, then I should be able to measure that. And the way that you did this was basically the nuclear ligation assay. Typically, you start by cross-linking DNA that would stabilize the chromatin in other ways. You can actually stabilize the chromatin in other ways, still works. And then basically follow the steps of the nuclear ligation assay. You cut and you ligate. The one change that you make is that you fill in the sticky end. So after you've cut with the restriction enzyme, you fill in the sticky end using biotinylated nucleotides. What that allows you to do is that after you've ligated, there's sort of boring bits that have pure blue sequence or pure red sequence, so they don't really reflect the ligation junctions. The ligation junctions, where the blue meets the red, where the two loci meet each other, are marked with the biotin, and so you can pull down those biotinylated ligation junctions and sequence them. The first time that we did this, we produced 4 ligation junctions, and I would actually kind of talk about them in the Simpson style. I would just put up the sequences of the 4 ligation junctions on the board or on the slide, and I would talk about what each of them was doing. But today we're able to make maps that have many, many billions of these ligation junctions from a single cell, or not from a single cell, from a single experiment. And so it's no longer possible to talk about each of the contacts individually, and so like with the Simpson slide, what we do is we create these 3D contact maps. So I just want to show you sort of what this data looks like so that you're familiar with it. Basically, the human genome comprises 23 pairs of chromosomes. Any bit of any of those 23 pairs of chromosomes can, in principle, form contacts with any other bit of any of those chromosomes, and in practice, that's what they do. And so what you end up seeing when you look at a Hi-C contact map is something that looks kind of like this. It's a little bit washed out. If it wasn't, you could see that sort of in the background, it's still a little bit pinkish. So there are some contacts between loci that are different chromosomes. What you can see very dramatically is that there are 23 squares along the diagonal of the Hi-C contact matrix. What might that be? Well, of course, the human genome has 23 pairs of chromosomes, and so what you're seeing is that there's an enhanced contact frequency between pairs of loci that are on the same chromosome, even if they're very far apart. Of course, at the highest resolutions at which one can interrogate them, these maps contain actually more pixels than Google Earth images of the surface of the Earth. And so it can be very, very hard to generate these kinds of images, to share these kinds of images, to analyze these kinds of images. And at a certain point, when it becomes really, really difficult to analyze data, it's actually prohibitive to reproduce what anybody else is doing or to check whether anything that people are reporting is actually true. This is a big problem in the field, and so we've created lots of tools to make this as transparent as possible. So for instance, if you scan this QR code, then it'll immediately take you to a website with a version of this figure on it, and you can kind of browse this figure and change the color maps and explore it further and see whether it really looks like what I say it looks like. This figure is relatively innocent, but you'll see I'll put some QR codes on some of our more interesting figures later, and you'll be able to in real time, if you're like bored with the talk, you'll be able to explore whether this stuff is true, and if you find that it's not, you can either raise your hand or write a communication to Cell or whatever you find most constructive. Anyway, so as I was saying, you can zoom into these matrices. So, as an example, let's zoom in here to chromosome 8. What you can see is the contact frequency is shown here, ranging from white, no contacts, to fully red. Here, the max is 700, although on different slides the max will change. What we're measuring is contacts, in this particular case, per 250 kilobases squared. So I apologize, I should have parentheses here, but basically it's all contacts between a 250 kilobase interval and another 250 kilobase interval of the genome at higher resolution. Those interval sizes get smaller and smaller as your matrix resolution gets higher and higher and higher. And what you can observe here as you're looking at the data is that loci that are very, very far apart have a relatively low contact frequency. That contact frequency gets higher and higher as they get closer in 1D and sort of skyrockets as they become adjacent in 1D or nearly adjacent in 1D, reflecting the fact that loci that are nearby in 1D tend to bump into one another quite frequently. This is true not only for this chromosome, but indeed you see this for every locus on every chromosome and every organism in which you've done a successful high-C experiment. It really reflects the underlying physics. At the same time, this isn't a monotonic function, so yes, indeed, on the average, contact frequencies tend to get higher as you're getting closer and closer in 1D. But you can see that there's a lot of variability in structure to this data, and that's exactly what we're going to talk about. Now, we're not going to have time during this talk to talk about all the different kinds of structures that you can observe in high-C matrices. What I really want to focus on is just showing how you can use high-C to carefully dissect a particular phenomenon, and that's the phenomenon that I introduced you to at the beginning, which is the phenomenon of how loops form in the genome. And what we'll specifically do is really, track the sort of effort to map these loops and understand these loops using high-C and other methods. Okay, so what should a loop look like in the human genome if formed by high-C? Well, you have two loci, A and B, that are forming a loop. So they should often be in contact. As you move away from these two looping loci in 1D, the contact frequency should decline. And so when a loop is present, you should observe a very bright local maximum in contact frequency within the matrix. So that was really one of the central goals of the initial high-C experiment, was to enable the mapping of loops by looking at where these local peaks were at. Unfortunately, the very first high-C matrices didn't allow you to do this. So here you can see the very first one that we published. At chromosome scale, it looks good. There's like enriched regions, there's depleted regions. It's all very nice. But when you look at finer scales, such as here, or here, and here I'm down at kilobase resolution, so every pixel corresponds to all contacts within kilobase, 1 kilobase of the genome, another kilobase of the genome. You can see that the contact frequency is is just sort of looks abysmal. The contact frequency matrix looks really abysmal. And the problem is, of course, genes are very small with respect to chromosomes. Genes are very small with respect to chromosomes, so if I want to understand how genetic regulation works, I need to be able to look at the matrix at this resolution, not just at this resolution. There were subsequent efforts to really improve the resolution of high-C matrices, but in general, the sort of gestalt was the same. They looked good at chromosome scale and looked very bad at kilobase scale. And then, due to some fantastic improvements to the protocol that were introduced by Suhas Rao and Elena Stamanova and Miriam Huntley and others on my team, Elena is actually here in the audience, we developed a refined version of the high-C protocol called in situ high-C. This was actually about 5 years after the publication of the first high-C paper. And the nice thing about in situ high-C is that when you combined it with enough sequencing, you could just keep zooming in on the matrix and it would just look better and better and better. And so here we are down at kilobase resolution, and all of a sudden you can see these very, very bright local maxima, exactly as you would expect. And so that's good. We can finally map the loops. And I do want to highlight that we've really tried to be as open as possible with our data. So when we first published this, this was actually more than, order of magnitude more data than all high-C maps that were published up to that time, and we shared it openly and interactively using these sort of interactive visualization tools. This was about half as much data as phase 2 of ENCODE, their large data release. So it's a massive amount of data just to map, just to create these kinds of high-C maps in about 10 cell types. The very striking thing that you observe when you generate this sort of data is that these very, very bright peaks, when you looked in 1D at the 1D genomic features that were present at these loci, overwhelmingly you saw a feature sort of again and again, which was you saw these binding sites that simultaneously bound CTCF, RAD21, and SMC3. Now, CTCF was thought to be an insulator protein. It was thought of as an insulator protein that defines regulatory environments in the genome. It had been long suspected as being involved in looping, so it was surprised to see it at nearly every loop. Nevertheless, it wasn't a total shock to see it involved in this phenomenon. It was more surprising on the whole to see RAD21 and SMC3. These are subunits of the cohesin complex, which is a sort of donut-shaped complex that was thought to be mostly involved in sister chromatid cohesion at the time. So we're very, very surprised to find that. Anyway, but the thing that was actually most shocking from all of this was the following fact. So CTCF doesn't bind to the genome willy-nilly. It tends to, well, it binds to either one strand or the other, because DNA is double-stranded, right? So this is sort of true with any protein that binds with one strand or the other. So that means that I have a pair of CTCF sites. They can either both be bound in the forward orientation, both be bound in the reverse orientation, be bound pointing away from one another, which we call the divergent orientation, or the motifs can point towards one another, which we call the convergent orientation. The striking thing is that when you look at the CTCF motifs bound to a particular loop, overwhelmingly they're bound in the convergent orientation. Now, this was really a shocking finding for us. I mean, I want to highlight, like, in this particular example, these two motifs are about 15 bases long, and they're separated by 100,000 base pairs. We were seeing this pattern whether they're separated by 100,000 base pairs, or a million base pairs, or even millions of base pairs. And it's kind of shocking how do these motifs figure out to point to one another over those distances. This was particularly hard to square with what was really the dominant model of loop formation at the time, which is loop formation by diffusion. The idea, and this emerged, been around for decades, was that the way loops form is that some looping protein lands in the genome and some other looping protein lands in the genome, and the genome sort of fluctuates, as that's sort of what it does microscopically. It's just fluctuating, and eventually these two looping proteins are very, very close to one another, so they decide to hold hands and form a loop. That's the model of loop formation by diffusion. I would say that pretty much everyone thought that this is how loops form at the time. And this model has this interesting property, which is that, whether or not it's a really good explanation, it was a pretty darn good explanation, but it's incredibly hard to falsify. There's very, very few behaviors that you could imagine observing that you couldn't somehow square with the diffusion model. And so this thing, was sort of, unkillable. But the thing is that diffusion actually works really, really, really badly with the convergent rule. And I want to just sort of give you guys a sense of scale. So if you blow everything up by a factor of 1,000,000, then the CTCF protein is about the size of a pea. And the chromatin interval that links a pair of looping CTCF proteins can be kilometers long. So how in the world do these two pea-sized proteins figure out that whether the kilometer of chromatin that separates them is sort of in that configuration or in that configuration? This was very, very, very hard to understand using the diffusion model, and yet we saw this configuration 300-fold more often than we saw this other configuration, the divergent configuration, just in the raw data. So it was really, really shocking, and we knew that there was a very, very big problem, and we really struggled to try to understand this, and so we started to speculate wildly. We call this a theory. Here's our theory. The theory is that the two cohesin rings two pairs of cohesin rings tethered to one another and land at adjacent sites on chromatin, and then the rings start to slide in opposite directions. Now, of course, when you're driving, you don't want to drive forever. Eventually, you want to stop. So these things, they want to stop. And I don't, you don't always stop, right? I like to stop for stop signs, but I only stop for a stop sign if it's pointing towards me, right? That's the traffic rule. If the stop sign doesn't point towards me, I won't stop. Whoops. Sorry about that. So what we can see here is these two. Okay, so you've got these cohesin rings, right? Because we knew cohesin was somehow involved. We knew it formed a ring. And they're sliding in opposite directions, and they're going to stop for a stop sign. Now, what could the possible stop sign be? Well, we thought maybe the CTCF motif serves as a stop sign. And if, in fact, it serves as a stop sign, then this subunit ought to be stopping, because it's seeing that CTCF pointing towards it. Now, this other cohesion ring is seeing a CTCF pointing the other direction, so it's not going to stop. Now, all of a sudden, it sees the CTCF pointing towards it. It's going to stop, too. If such a machine, which we dubbed an extrusion complex, existed and behaved in that sort of way, then it would tend to form chromatin loops between pairs of loci in the convergent orientation, and that was very, very exciting. The one thing that is very, very important to bear in mind is, despite us having this, sort of Technicolor video, we didn't really have any evidence whatsoever to support this model, and indeed most of the extant chromatin models directly contradict this in various ways. So we really didn't feel very comfortable talking about this. And so we said, well, let's try to come up with some more evidence. Let's put our physics hats on and simulate what were to happen if you put chromatin into baths of these extrusion complexes. So we put together a very, very simple coarse grain model for chromatin in sort of a bath of these extrusion complexes, and then we started to try to predict the results of experiments. So let me show you some of the experiments that we did. So here on the left, I'm always going to show you predictions, and on the right, I'm going to show you actually experimental high-C data. This particular locus is what we call a transitive triple. This is something you very commonly see in high-C data. So this region comprises three loci, A, B, and C. A forms loops with B, A forms a loop with C, and B forms a loop with C. So according to the convergent rule, then you would expect that you should have a forward-oriented motif at A, because it's looping to two downstream loci, a reverse-oriented motif at C, because it's looping to two upstream loci, and motifs in both orientations at B, because B has to loop both to upstream and to downstream loci. Okay, so we knew that, and so we did some simulations, and the simulations matched the data, but this wasn't actually a prediction in the strictest sense, because we already sort of knew what the data looked like, and we were very nervous. We wanted to do predictions in the strictest sense. So the predictions that I show you actually in the rest of this slide are actually predictions that we did chronologically prior to the corresponding experiments. What we started to do was we started to look at these various motifs and try to disrupt them in different ways and see what happened. So first, and we did very targeted sort of CRISPR disruption, so we're taking out as little as a single base and at most 100 base pairs. We're very targeted at very little collateral damage. So what, for instance, might you imagine would happen to these loops? Just for simplicity, let's talk about just a loop from A to B, a loop from A to C, a loop from B to C. What do you think would happen to loops if we got rid of the CTCF motif located at A? Yeah, so Bridget, what's your name? Alexander. So Alexander's prediction is that you would see loops only between B and C, because the CTCF motif at A is responsible for loops anchored at A, and terrific. That prediction, in fact, is exactly what our simulations did, and is exactly what we observe in the data. So we were very, very happy about that. But that's actually not a test of the convergent rule. That's really just a test of whether the CTCF motif actually anchors the loop. And while we're very happy to see that this very 20 base pair motif that we targeted, in fact, was responsible for these changes spanning about 800 kb or 600 kb or so in this region. So we're very happy about that. But it wasn't actually testing the convergent rule. It wasn't testing whether the orientation mattered. So the next experiment that we did is we got rid of the B reverse motif. So what would you expect might happen to get rid of the B reverse motif? So one prediction is you should retain the loop from A to C. Any other predictions? So, okay, so the prediction then is that you get a single big loop
from A to C. Well, actually, our simulations predicted something similar, right? We predicted that you'd preserve that loop from A to C, but you'd also retain the loop from B to C, right? Because the loop from A to B is anchored at the reverse-oriented CTCF, right? If you don't have that reverse-oriented CTCF, you're not going to have a convergent pair, and so the loop from A to B won't form. But the loop from B to C will still form because you're still going to have that forward motif at B, right, and the reverse motif at C. So we predicted that you'd retain A to B and B to C, and in fact, that's what we observed. Do people see why that's the prediction that we made, that why we predicted specifically that the B to C loop would be retained?
So what did you remove again?
We removed the B reverse motif. So this is the, sorry if this is unclear. So these two motifs, one is facing this way, and one is facing that way. So we removed the one that's pointing in reverse. I'm sorry if that was unclear in the projection. So does that make sense? Terrific. Okay, well, you have good news, which is that you have another chance to demonstrate your complete mastery of the rules of looping, which is that we then removed the B forward motif. So what would you predict would happen if we removed the B forward motif?
The A will remain, and the A to C will remain.
Yes, the A to C will remain, and which other loop will remain?
The A to B, they're both convergent.
That's right. So the A to B will remain, the A to C will remain, but the B to C will disappear. And in fact, that's exactly what we observe. I do want to highlight this experiment was a little bit messy. But we also got another chance to sort of redeem ourselves. This experiment came out more cleanly. And we're doing these experiments, we realized that other people were thinking about these ideas, so we didn't have time to do that experiment over and over again. But now what I want to highlight is this very, very striking behavior that we just observed in the last two experiments, right? Which is that the B reverse motif anchors the A to B loop, right? And the B forward motif anchors the B to C loop. So the orientation really matters. Okay, so then we said, let's probe whether the orientation matters another way. Why don't we take the B forward motif, instead, we're not going to harm any base pairs. All the base pairs in the genome are going to remain. All we're going to do is we're going to take the 20 base pairs of the B forward motif and we're going to flip them around. So now it's going to be reoriented, but pointing backwards. So what would you predict would happen? Yeah, so what loops would you expect to see after you've done this change? That's right. So you'd expect to see AC and BC. And what's really, really striking about this is that the prediction is that if you invert the B forward motif, it's the same from the standpoint of these two loops as really deleting it. And that's the extent to which orientation matters, right? We're not getting rid of any bases, but we're changing the orientation of 20 bases, and we expect to lose a loop just as though we deleted B forward, and in fact, that's exactly what we observe. Finally, it's interesting to note that once one has deleted B reverse, the B forward motif becomes like a flip-flop, right? When it points forward, you would get B to C, but if you flipped it around so it pointed in reverse, you would expect to simultaneously lose the loop from B to C and rescue the loop from A to B. And of course, that's what our physical simulations predicted, and that's what we observed in practice. So at this point, we felt that we'd done a series of experiments that probed the convergent rule, that probed whether this was really how things were going on, and that confirmed some key predictions of the extrusion model. And so we published it. I do want to highlight that there were a number of other groups working on this at that time, so there was a terrific, while our paper was actually under review, there was a terrific preview that appeared in Cell, perspective piece that appeared in Cell, by Mike Nichols and Victor Corses that highlighted the fact that the convergent rules suggested an extrusion model. And similarly, there was a bioarchive piece from the Murray group that appeared shortly thereafter, which also proposed such a model. And then our paper came out shortly thereafter. We also had a paper a few months later showing that you could do much, much more ambitious engineering experiments based on these principles that actually rearrange whole chromosomes. So we were very excited about all of this, and in fact, many groups became kind of excited about this model, and so there's a lot of work now studying loop extrusion. I also want to highlight that as we were doing these experiments, we found a really, really terrific review by Kim Naismith, who's one of the pioneers of the cohesin field, and who had proposed in 2001 that perhaps SMC family complexes, including cohesin or such as cohesin, could form loops by extrusion during interphase, during metaphase, not during interphase, but during metaphase. And that actually made us feel much better about the model, because if Kim Naismith thought that this was not crazy, being a pioneer of cohesin and all, then maybe it wasn't crazy for us to say it either. So anyway, suffice to say that there's been a lot of enthusiasm about this. And so what I want to show you, and so where are we at? So now we're able to map the loops, and we're able to actually think about what is the mechanism of their formation. So we were excited about both of those things. And so in the remaining sections, what I want to get into is that we can actually study these loops today, both through space and over time. So let me tell you about our efforts to study loops over time. And I do want to highlight that after the proposal of the loop extrusion model, there were many different groups who did many things to try to confirm the loop extrusion model. And there are many beautiful confirmation experiments. I think this is a particularly nifty confirmation experiment, but it's one example of many. There's just not enough time in this talk to go through all the different forms of confirmation that our group did and that many other groups have done. Okay, so in the previous series of experiments I showed you, we were playing around with the CTCF motif, but of course we knew that based on this hypothesis, if we disrupted the entire extrusion complex by degrading the cohesin ring, we expected that we should be able to totally destroy all of these loops that we were observing, and we spent actually years trying to find a system where we could very reliably degrade nearly all of the cohesin complex or any of its partners, and finally we actually found this terrific Degron system published by the Kanamaki group in Japan. And when we tested it, we found that actually it overwhelmingly got rid of cohesin, and so we're very happy because we could finally do our experiment. And here's the experiment. I do want to highlight. So the loops typically appear at the corner of these bright domains where all pairs of loci have enhanced contact frequency. So in the HCT116 RAD21 Degron system, this is a system which, when exposed to plant hormone auxin, rapidly degrades all the RAD21. So prior to treatment with auxin, when you generate high Cmaps, you see just our automated algorithms annotate over 3,000 of these loop domains. After 6 hours after treatment with auxin, every single loop domain is gone. The algorithms actually annotate still go down from 3,300 and something to 9, but if you look at the 9, you can see that they're actually not real loop domains. They're anomalies due to factors I'm going to talk about in the second talk. So they all go away. Anyway, if you don't believe that, you can scan this QR code and browse around and demonstrate that this isn't just an artifact. The color map I chose, and you can browse around the genome and try to find other loci as well. And well, I don't think you will find any loop domains. I would be amazed and concerned if you did. Anyway, I do want to highlight that there were a number of other papers along these lines that appeared all roughly at the same time. I have to say that I think the most innovative of these is the work of Elfedja and Nora, and I say that for a very specific reason, which is that Elfedja and Nora, many of the groups that were trying to do this sort of thing, yes? What happened to cells? That's great. So this is a terrific question. They can't go through metaphase, so they start to rapidly get stuck in metaphase. So that's a terrific question.
Did they die? Did they die?
Yeah, eventually they die. But they will last long enough to do these sorts of experiments. But yes, eventually they'll die. We have been doing versions of this experiment where the cells are arrested in G0, and there they seem to be able to survive much better, but we haven't redone all of these experiments in that circumstance. Or we haven't redone all these experiments after long lives in that condition. But I do want to highlight, Elfedja and Nora figured out something really important, which is that many groups were successfully reducing CTCF and cohesin and these other factors by a significant amount. But he realized that a significant amount, and not seeing much, and he realized that a significant amount wasn't enough, that all of these features would remain even if you got rid of like 80% of the expression of proteins, so you really need to get 90%, 95% of the protein away. And then once you realize that, I think multiple groups, including ours, were able to understand why these experiments had not been working for years, in fact. This was actually kind of the one experiment that I thought we would be able to get to work years earlier, and it kept failing, and that was frustrating, but we decided to publish our other stuff anyway. But anyway, it finally worked, so that was good. Now, moreover, the really nice thing about this system, which was super exciting, is because we're treating auxin, we could control the level of cohesin really nicely, but we could actually withdraw the auxin and watch all the cohesin come back, and the cohesin came back very rapidly. And so what you're seeing here is the auxin degradation and then recovery over a 3-hour period. And so you can see that all the loop domains go away, although some of the domains still remain, but those aren't loop domains. Right? All of the loops go away, and then very rapidly the loops come back. And what was nice about this was that it allowed us to measure the rates at which the loops reformed across the genome. And this had very striking results. So for instance, this loop is 900 kilobases long, but it largely reforms after 40 minutes. So that tells you by long division that loop extrusion, if it is real, is happening at least 400 base pairs per second. This was quite important because people had suspected that, for instance, maybe polymerase is pushing the extrusion complex around, but the fastest known polymerases are sliding about 100 base pairs per second, not fast enough to explain this behavior. So it's very, very fast. In fact, we actually think this is a lower bound, and the real speed may be faster, and we're working on exploring that. So we could explore loops through time, and finally I want to close by noting that it's now actually possible to explore loops through space. A very basic question, I think, that we started with was really the power of the microscope. And a very reasonable thing you might ask is, well, it's sort of fun to go into this high-C world with these funny matrices, with all these claims about what they mean. But do the maps accurately reflect 3D genome architecture via a completely orthogonal method, via what I would venture to say is really the gold standard for understanding structure, namely when you look under the microscope? And there were some really terrific papers. I'm sorry, this is Science in 2018. So really terrific papers that appeared in 2018. One by a group we collaborated with Ting Wu, Marty Renam, really led by Ting Wu, a brilliant microscopist at Harvard, and the other group led by Xiao Wei and Alistair Bodeger, that developed the following sort of technique. So for a long time it was possible to illuminate particular loci in the genome using fluorophores, using what they call FISH, fluorescent in situ hybridization, where we designed probes that bind particular bits of the genome and fluoresce. So what both groups developed was a method that allowed you to actually wash the probes off and then rehybridize a new set of probes. So what that meant is that instead of looking at one or two loci and using one or two different colors, you could use a single color, but you could hybridize to one locus, and to another locus, and to a third locus, and to a fourth, a fifth, a sixth, etc. You could just keep going dozens and dozens of times. And in that way, you could figure out the location of locus 1, and then locus 2, and then locus 3, and then locus 4, and you could produce data that looked like this. This is an example of real data, tracing 9 different adjacent loci on one chromosome and on the other chromosome in real nuclei and in single cells. And I just want to show that this actually produces, you could ask, well, does this stuff look at all like high-C data? So what I'm highlighting, this is actually data that we've had nothing to do with, didn't generate the data, didn't do any of the analysis. This is from the other paper from Bintu et al. It's a really beautiful paper. So on the right, you're looking at high-C data that I've been showing you, although it's a little bit more coarse-grained because the resolution was sort of lower than ours. They're working to improve the resolution. I'm sure that's going to happen, but so far, the resolution's a bit lower. So this is at 30 kb. Each pixel is all convex between 30 kb and 30 kb. So high-C data on the right. And on the left, this is showing the fraction of cells where a pair of loci are within 150 nanometers from one another based on these 3D traces. And I want to highlight that this particular threshold is not particularly important. You get roughly the same answer for a pretty broad range of thresholds around 150, 200, 100, what have you. And if you look at these, they look pretty much identical. And you can see these bright peaks corresponding to loops, and you can see the whole loop domains, and you can see basically all the features of the high-C data are beautifully recapitulated in the microscopy data. I do want to highlight that the microscopy data it has a disadvantage, which is critical, which is that you can only visualize relatively short intervals of the genome and at relatively coarse resolutions. But of course, it has a spectacular advantage, which is that instead of having a picture that looks like this, all of a sudden we could visualize individual loops in space. And so this loop here from actually PGP1 cells, so it's a lymphoblastoid cell, a fibroblast cell type. So here are the two loop anchors, and then the loop it traces out, visualized in 3D. Or another example of a different loop, here's the two loop anchors, and then you can sort of trace out their whole interior in 3D. And so it became possible not just to visualize the loops in time, but to visualize them, to trace them over time, but to explore them in space as well. Finally, just in closing, I want to highlight that all of our methods are publicly available. So our in situ high-C protocol is public. It's a standard protocol for both the ENCODE and the 40 nucleome project. We've made lots of software available to help other people do these experiments. So for instance, the juicer pipeline will take your raw sequencing data from high-C and annotate all of the loops and create these matrices. The JuiceBox software allows you to explore all of these matrices. There's now versions of this on the web, which you could use with those QR codes that I was showing, which allows you to do this stuff on the web. And in fact, if you have any, you just put your files in Dropbox, and that can become a permanent server for a sort of web-based browser. You can send links to your friends. We rendered about a trillionth of one of these maps in virtual reality. You can download this at Google Play. These things that look kind of like Mount Doom from the Lord of the Rings, those are the loops. There is a little bit of smoothing. This is the only matrix I've shown you today that has smoothing. A little bit of smoothing here. The things that look like the Eye of Sauron are the loop calls from our automated loop caller, so you can use this to sort of validate your loop calls. And finally, these things that look like the wall from the Game of Thrones, no spoilers, please. Those are that bright diagonal, the really, really high contact frequencies between pairs of nearby loci. If you download these, I suggest that you look straight up, which will allow you to jump around the landscape, which is a great way to develop intuition for what high-C maps look like. And then in the next talk, I'm going to focus on some of the other tools that we have, including 3D DNA and JuiceBox assembly tools, which facilitate genome assembly. Finally, if you have questions, there's hundreds of members of our online forums that ask questions about the experiments and the software and the analyses. And you can join and ask questions, and we try to answer them as best we can. Just for those of you who are curious about when will this be coming to my cell type or the system that I care about, we are working on a high-C atlas of the human body so that everyone can have high-quality high-C data in whatever system they're interested in, or a very wide variety of tissues and hematopoietic cell types as part of the ENCODE project. All this work is funded by all of the following funders. I do want to highlight there's many collaborating groups that we've been involved with, especially the microscopy work together with Ting Wu and Mark Marty Renom's lab, Guy Neer and Irena Farabella, the co-first authors from there, as well as Cynthia Perez-Estrada from my group. And I do want to highlight that all of the sort of key concepts we developed as part of the Center for Theoretical Biological Physics grant to my lab and several others in Houston, and the nuclear architecture work that I showed you just now. The key trainees are really terrific. Adrian Sandberg and Sue Oscar out here helping to develop all of these sorts of key ideas. And I really want to highlight Sue Oscar, who's been the sort of main driver of all this, a really, really brilliant trainee. And then Cynthia Perez-Estrada, as I mentioned, was the lead on microscopy work. And if you'd like a job, we're hiring postdocs both in Houston and in Shanghai, so let me know. Thank you guys so much. I guess there's probably, I don't know, if maybe we'll do a few questions now, and then I'll tell you about other stuff. Yes?
So, are the CTCFs leaky, or the TF binding sites leaky with a pair conversion and then another pair conversion outside of it? Do some of them leak through?
So that's a great question. I would say that right now we think that they are. I mean, I can't rule out. The thing that becomes a little difficult is it's difficult to distinguish between a leaky CTCF site because there's two senses in which a CTCF site can be leaky. It can be leaky because it's not always bound by CTCF, or it can be leaky because even though it's bound predominantly by CTCF, even when things hit the CTCF and arrest, the CTCF sometimes falls off or allows it to pass. And it's hard to distinguish between those leakiness modalities, but it's completely clear that they are leaky in at least one of those senses, I'll say.
And by leaky, we mean like the outside one would be as bright or just there?
That's a terrific question. In theory, we would think that the outside one would be less bright, but I do want to say that in practice, decisive experiments where you can observe that effect have not happened yet. So the models sort of suggest that kind of thing ought to happen, but it's, there isn't really terrific data. Yes?
Have you tried doing cookies in CTCF or some active effects, things like this, on this drone system? Or?
Yeah, so we're working on that sort of stuff right now.
Okay.
Yes? Yeah, so that's a terrific question. We were really, really afraid of that question, actually. I'll give you a simple example, right? Here's a great example of a protein that you should be worried about, which is the histone protein. It's a nucleosome every couple hundred base pairs. How in the world is cohesin? Cohesin was not known. It was known in vitro that it could, in principle, slide along DNA. The notion that cohesin could slide on chromatin was not known when we proposed the model, and yet what we were suggesting was that it could slide across megabases of chromatin. So literally, it was sliding across thousands and thousands of nucleosomes for this model to be true. And that was actually really concerning. We didn't know whether we'd be able to do that. Ultimately, and we can get into I showed you some of the evidence here, get into some of the other experiments we did along these lines. Ultimately, proposed the model. Subsequently, there's been some really nice experiments showing on DNA curtains and chromatin curtains showing that cohesin can in fact slide along nucleosomes, which is terrific. How does it interact with every single other protein that's CAF? That's a great question. We don't really understand it, and it's something that we think about a lot. Yes? Yes. Yeah, in the absence of cohesin, these things cannot go through the cell cycle. So yeah, it's within one cell cycle. We also did a version of this experiment, I was kind of alluded to it before, where we did this with synchronized cells. The results were the same. Although we didn't do it as many time steps, but the general results were the same. Yeah, so one of the things that we observe in these experiments is that the ubiquitination process will happen to the bound cohesin, such that even if it is engaged on chromatin, it will fall off. Rad21 will be degraded. Yes. I do want to highlight that there's one other interpretation of the data that I've shown you, which is that the loops are constantly falling off and reforming, et cetera, and that's happening, but I wanted to answer some of the other questions, so I won't get into it right now, but it's clear from other experiments that we've done that the lifetime of these loops is very long in a way that is inconsistent with that, but that's actually a big, kettle of fish, because there's a lot of data about that, and I won't get into all of it, but our take is that it is definitely the case, that it's not just spontaneously falling off. Yes? Sure. Sure, yeah, absolutely. So terrific question about the dynamics. So I'll tell you that the experiment that we've done in this space, approach to dynamics, is, let's see, so, yeah, so let me show you this experiment, which I think is very, very helpful. So here what we do is we treat with auxin and something else, and then we see how it affects the recovery process. We treat with auxin, we lose all the loops because all the cohesin is gone, right? So you can see here, untreated with auxin treatment, all the loops are gone. And then, of course, with no drug, the loops recover after we withdraw the auxin. But if we inhibit transcription, actually, and one of the ideas was that RNA polymerase was responsible for pushing the extrusion complex around. That's actually rebutted by this, because you see, even if you inhibit transcription, the loops reform. If you inhibit replication, the loops reform. But if you inhibit ATP, the loops don't reform. If you inhibit ATP synthesis. So what that tells you is that either cohesin loading or cohesin sliding or both require ATP. Now that's actually very important to be able to do this experiment, because in the presence of this experiment, what this experiment tells us that in the absence of ATP, new loops will not form. And so then you can do sort of a version of this where you inhibit ATP synthesis, but you don't mess with cohesin at all. And in that system, you know that if you believe all of this stuff, then you know that new loops aren't forming. So the loops that you have got to kind of hold on for dear life because they're not going to get replaced. So the system's no longer at equilibrium. And so then what you can do is sample at various time points and see if you observe any decay or any weakening of the looping signal. And we've done this as far as 24 hours out. We'd like to do this further out. We haven't done it yet. Done this as far as 24 hours out, and I do want to highlight that we see no appreciable weakening of the looping signal. So our interpretation of that data is that the half-life of the loops when they are fully formed is of the order days. And I do want to highlight that there are other groups that have measured the half-life and the binding time of cohesin and CPCF and have estimated numbers that are much smaller. My interpretation is that there are many subpopulations, and that the subpopulation that actually has loops has a much longer lifetime. The other subpopulations indeed have a shorter lifetime. I think that enables one to synthesize all the data in a consistent way. So what we think is that once these loops form, they stay that way for long periods of time, days. So yeah, that was question one. We use what? What's that? So yeah, so in the model that I showed, yeah, you can see I actually showed like this little video which shows how we're modeling this. So you have two cohesin rings. I realize that this is super controversial and that we had, I'll be just frank, we had no intellectual basis whatsoever to choose two, other than we had to choose some number, and two is some number.
There's a reason you want to have
I mean, I don't want to get into stuff that's like above my pay grade. The cohesinologists understand these issues much, much better than I do. We needed a number, and we were in a rush, and two seemed good. Yeah, so maybe one last question. And then I'll maybe go on to the next section just so we don't fall super behind. Is that, or should we break? Yeah, no, we can break. Is that? Yeah, no, whatever. Yeah, you're in charge. I mean, you tell me what to do. Oh, terrific. Yes. No, that sounds good. Yeah, okay, that sounds good. Are we? Do I have time? How many questions would you recommend that I take?
One more?
One more. Okay, terrific. Sounds good.
So, in your predictions,
Yes. Yeah, that's a terrific question. So let's see if I have my super handy backup slide for this particular question. It's here somewhere in one of these slide decks. This information is located. Oh gosh. Okay, I don't see it in this particular deck. So I'll quickly explain why this is a terrific question, and then how we think about this. But I won't make you suffer through me trying to find where in the 700 slides that I have on this I have the relevant slide. Okay. So this is a really good question. So what's being pointed out is the following, right? There are two features that are visible in these maps. There are loops, which link pairs of loci, and domain boundaries, right? We see this sort of like liney feature, which suggests that sort of the upstream and downstream regions have some diminished ability to form contacts with one another. And these are domain boundaries. I do want to highlight that a loop is a pairwise feature that reflects the linking of a pair of loci. And a domain boundary is actually a linear feature. It just exists at a position. And the point that's being made is that sometimes when the loop goes away, the domain boundary doesn't. And gee, isn't that really weird? And I do want to highlight that actually our model predicted all of this. Although I didn't have time to talk about it because I didn't want to take too long, but it's a great question. And I do want to also highlight further that it's not always the case that the domain boundary stays, right? Sometimes it stays and sometimes it doesn't. So for instance, when we disrupted the loop anchor at A, you can see that the domain boundary at A disappears. But in all of these subsequent experiments at B, the domain boundary remains. And I want to highlight that one of the predictions actually of the extrusion model, as soon as this was actually one of these cases where writing out, the simulations was helpful to us in really thinking through the cases, which is that you realize that you need to have a behavior for the following case, which is what happens when CTCF is cohesive. And you know that you have some dynamics for cohesin to land and cohesin to fall off, and for cohesin that bumps into CTCF, but you also need a behavior for when cohesins run into one another, right? Because otherwise, it does not compute. So we said, well, what could that behavior possibly be? And there were really two possibilities. Consider one is that the cohesins stall, and the other is that one of the cohesins falls off. And we said, well, look, if it's the case that the cohesins stall, then I have my then I have my CTCF and my cohesin lands and sort of binds to it. And it obeys this sort of inward orientation preference, which we gives rise to convergent rule. But then shortly thereafter, another cohesin is going to come from the other direction and bump into the first cohesin, and it's going to rest. So what you end up having is cohesins are resting on both sides of CTCF motif, which would efface the convergent rule, and you'd have tons of loops, divergent motifs, and forward and reverse, etc. It just wouldn't, orientation would matter a lot less, or wouldn't matter at all in such a model. So we said that is actually not consistent with our data, and what we think is probably happening is the cohesins are falling off. And we hypothesized actually this factor called waffles unloading them. And there's beautiful work by Harhuis and others in Cell in 2017, which appeared in one of these slides, which actually kind of confirmed that this happens, or was entirely consistent with this set of predictions. Now, the net result of this is that cohesins, so the prediction is that cohesins, when they hit an arrested cohesin in CTCF, other cohesins will fall off. And so when you think about what these contact domains represent, what they represent is really the span of a set of cohesins, right? Cohesin, an extrusion complex that lands anywhere here, will sort of naturally slide out to the boundaries, but will face some sort of impediment when crossing these boundaries. Right? That's really what the domain boundary represents. And the critical point here is that a cohesin represents an impediment to other cohesins, regardless of its orientation. So, as long as a loop anchor remains at a locus, that locus will be a domain boundary, no matter which orientation that loop anchor is in. So here, when you got rid of the loop anchor at A, there were no other CTCFs. So the domain boundary goes away, right? But all of these disruptions at B left at least a single loop anchor at B, and so the domain boundary remains. And the slide I was looking for, an experiment that we published also, and which we consider a crucial experiment because of the importance of this question, which is there's a very strong prediction, which is that if you were to get rid of both of the anchors at B, you would merge the domains. And in fact, that is what we observed when we did such an experiment. Unfortunately, I don't have it on this slide. But it's a terrific question. It highlights a very important thing, which is that the rules for domain formation and the rules for loop formation are simply different. Extrusion will form, so first I want to highlight, domains can form via methods that have nothing to do with extrusion, as we showed in this sort of slide where we got rid of cohesin and you had all these nice domains to the left, or this really bright domain to the left. But it's also the case that even within the extrusion model, domains will form between pairs of CTCF sites regardless of orientation. Loops only form between pairs of CTCF sites in the convergent orientation. Yeah, they certainly can be, and they can also be, in some cases, smaller than loops. But the key thing is that extrusion domains obey different rules than extrusion loops. So this is a very subtle but critical question in thinking about the origin of domains and loops. Please let me know. I'm told that this may have stopped working in the previous talk, but no one let me know. Those of you in the back, I'm looking at you. Although you were like, wow, we finally enjoyed some restful silence. Anyway, I realized that, one talk is okay, but two talks can be very, very difficult. So I promise that this talk will have far more cuddly animal pictures than the last one. So, I mean, there's a whole internet full of people who watch cat pictures all day. And you guys can do it as part of legitimate work. So please take the opportunity. So what I'm going to tell you about is I want to highlight that Hi-C and the methods of 3D genomics more generally can answer problems that are specifically about nuclear architecture in powerful ways that I hope you saw in the previous talk. But they can also resolve problems that have seemingly little to do with nuclear architecture, but that can be quite important. And so that's going to be the focus of this talk. And specifically what I'm going to focus on is the problem of genome assembly. So let me describe this to you. So people have been trying to assemble genomes for a while. The first time a mammalian genome was assembled was the Human Genome Project. That took like 30 years and $4 billion. Things have at least improved now to the point where you can generate like lots of reads, typically short reads, very, very cheaply. And then there's this thing that you can do that's kind of inexpensive, which you can take these reads and you can map them back to a reference genome. So if you have a reference, once you've spent your $4 billion on your human genome reference, you can just take your reads and align them back to your human genome reference, and you can notice little changes sometimes. So if there's like a single base pair that has changed, or 5 base pairs that have changed, or 20 base pairs that have been inverted, you can notice all these little changes. And this costs actually about $1,000 to do this kind of DNA resequencing. So sometimes you may have heard about like the $1,000 genome, and that Illumina, the sequencing company, has enabled the $1,000 genome. This is what they're talking about. They're talking about DNA resequencing. I have a reference for the species, but I'm trying to look for like little changes at particular positions. I see a little change, and then I annotate that little change. And so what I end up with is this variant list that can go on top of my reference. And doing this inexpensively is basically a solved problem. But there's this other question, which is, what if I don't have a high-quality reference in my species? So yeah, the human genome, I'm not going to have this problem ever. But what if I don't have a high-quality reference in my species? Or suppose I do have a high-quality reference in my species, but I want to be able to be sort of sensitive to all kinds of changes, no matter what the scale, right? In which case, yeah, maybe I want to take a patient and actually do their genome from scratch, not make the same assumptions that were built into the Human Genome Project and the subsequent references. So is it possible to create an assembly de novo, right, without having a reference for a species, or without using a reference for a species to create a reference in an inexpensive way? And for instance, gee, it would be really nice to be able to do it for $1,000 or a couple $100, and certainly we don't want to spend $4 billion on it. So that's right out. So what I'm going to do is just kind of give you a bit of a brief overview of how people go around assembling genomes, how this works, and just a tremendous summary. So the way that you do this is you start by producing a bunch of reads from your target genome, and then you sort of overlap these reads one on top of the other, and you can see that the reads often overlap a great deal, and so you can kind of make longer and longer sequences that can be much longer than the length of your read, and the result is called a contig, right? So this is, the Human Genome Project did this. Every genome project does this. This is how you make a genome, is you start by tiling your reads and making contigs. Or even if you chronologically don't start that way, in a certain sense you do logically start that way. So the problem is that well, this might work for a very simple and small genome, but this does not work for complex eukaryotic genomes. And the reason for that is that complex eukaryotic genomes are loaded with repeat elements. So this is like a sign element or a line element. It can be thousands of base pairs, it can be hundreds of base pairs, or thousands of base pairs, or tens of thousands of base pairs, or even hundreds of thousands of base pairs, or even longer. So these repeat elements, because they're repetitive, there's no way to tile across them, because you don't know, as soon as I get into the repeat, well, many of my contigs get into that sort of repeat, and I don't know when I exit the repeat which of the contigs sort of exits at that position. So everything breaks down at the repeats. And so the contigs that you get are much shorter than chromosomes. So, typically if you're doing like a short read assembly, the best assemblers around will produce hundreds of thousands of short contigs, whereas what you want is 46 long chromosomes. That's what you want, you want a single sequence per chromosome. And even with long reads today, you still end up with thousands of contigs, so some far more than the chromosomes, and it costs you a lot more money. So what do you do with all of these contigs, given that they're much, much shorter than the chromosomes? What you want to then do is to order and orient the contigs. That's how you make a genome, is you order and orient the contigs so that you can basically say, okay, here's chromosome 1. Chromosome 1 starts with this contig, and then this contig comes next, and that contig comes next, and that contig comes next, but I have to flip it around, et cetera, et cetera. And the way that you order the contigs is using what I'll just call generally linking data, right? There's many different kinds of linking data, which we'll get into. But the basic idea is you take a pair of contigs and say, you know what, I figured out with my linking data that these contigs are adjacent in the following sort of way. I hope you notice, right? I had to flip one of the contigs in order to do that. They're adjacent. There might be some repeat in between them. I don't know for now what that repeat is. But I am able to link the pair of contigs. And the idea is that if you link all of the contigs, perfectly well, then you'll end up with a scaffold that runs from one end of your chromosome to the other end of your chromosome. This scaffolding process, ordering and orienting the contigs along the chromosomes, has in the past required multi-institutional consortia, huge amounts of money. This is actually when the Human Genome Project was sort of at work, this is actually most of what they did was the work of scaffolding. It took the most time and the most energy, actually. So it costs huge amounts of money up till very, very recently. And so if you wanted to do a good job of scaffolding, typically you have, the resulting papers cost, millions and millions of dollars and have hundreds of authors. I should say that even with that, these scaffolds often fall short of chromosomes, despite being very, very costly. So what I want to tell you now is that actually Hi-C is a very, very powerful source of linking data. And for that reason, it becomes actually very, very relevant to the problem of genome assembly. So let's look at the data. Let's look at the Hi-C data again. Right, you can see, as soon as you look at the data, you start to realize why this is useful. Right away, we notice that there's an enhanced contact frequency linking pairs of contigs that are on the same chromosome. So the very, very first step in scaffolding, many people actually think of it as a separate step, but we'll sort of simplify, is anchoring. So you want to figure out this contig is from chromosome 1, and this contig is from chromosome 2, and this contig is from chromosome 7. And in general, these 5 contigs all come from the same chromosome, so I better try to figure out their order and orientation with respect to one another. That's the first step. In genome assembly, you immediately realize, gosh, Hi-C data is going to be really useful for this, because I'm going to see that elevated contact frequency whenever pairs of loci are on the same chromosome. And moreover, as you guys recall, when you zoom in, the contact frequency tends to increase the closer a pair of loci are to one another. Right? It gets higher and higher and higher until, as we said before, it skyrockets as you're close to the diagonal. When a pair of loci are extremely close in 1D, they have a very, very elevated contact frequency with respect to one another. So that tells you that not only can I use Hi-C data for anchoring, figuring out which pairs of contigs come from the same chromosome, but I can also use it to figure out whether the contigs are close or far. So to solve that ordering and orientation problem on my chromosome. And it was pretty obvious really from the outset with HiC, even before the paper was published, that you could do this. And indeed, many proof of principle papers that were very lovely came out sort of highlighting that you could in principle do this. And the way that these proof of principle papers worked is that they would take a genome, a known genome, known chromosomes, cut it into little bits, and demonstrate that you could reconstruct the correct ordering and orientation of those bits from using the HiC data. So a known genome was reassembled again, which is a very important, proof of principle. But of course, as we know, when you're solving a jigsaw puzzle, it is easier to solve the jigsaw puzzle if you sort of know what the answer is, and you kind of can look at the lid. What remained difficult to do was actually to be able to do this when you had no idea what the target genome was supposed to look like on data that wasn't simulated by just chopping the right answer into pieces, but real data that came from real contigging processes. And at the very end of 2016 and early 2017, you actually started to see some papers that were starting to use HiC data in this way. So terrific genome of barley appeared in early 2017, and xenopus late 2016, where they used HiC data together with many, many other data types. These are still these kind of 100-author plus papers. Many millions of dollars are spent. And they use many, many forms of linking data, but HiC is being used now to assemble new chromosome length scaffolds where they were not previously known. So this sort of started to happen in 2016 and 2017. But I want to highlight that, as of mid-2016, there was really no cost-effective strategy for solving these kinds of problems. This is highlighted. You can see actually there's the problem on the front page of the New York Times, the problem of the Aedes aegypti genome. Appeared Aedes aegypti as the principal vector of the Zika virus. There was a massive Zika outbreak. And the state of the Aedes aegypti, the mosquito, which is its principal vector, was actually displayed on the front page of the New York Times. It's probably the only time the scaffolding problem has appeared quite so explicitly in that forum. And people didn't know how to put these pieces together. In fact, Aedes aegypti has 3 chromosomes. So all these pieces were supposed to come together in only 3 chromosomes, and people didn't know how to do that. And it highlights the fact that, in the setting of an urgent outbreak like Zika, you don't have, years or decades and millions of dollars to sort out these problems. You want to be able to sort these problems out quickly. Okay, so now I want to tell you about a tool that we developed in my group, and all of this work I'm going to tell you about was led by a really brilliant postdoc, Olga Dubchenko, in the group, who's really, really wonderful and worked incredibly hard, and was able to work with members of the team to solve both the experimental problems that underlie solving scaffolding reliably, and also the very, very, very significant computational challenges that underlie using this data type to reliably scaffold genomes. So, in order to tell you about this, what I want to do is start showing you features of HiC data that you didn't see in the previous talk. So in the previous talk, we saw these domains, and we saw these checkerboard patterns, these bright peaks, et cetera. But there are certain things that you see in HiC map that HiC maps can kind of reveal to you that look very, very different than those features, and I want to tell you about them. So what I'm showing you here is a pretty crummy genome assembly. This is actually 2 chromosomes from the human genome, but they haven't been assembled very well. So what you're seeing is you're seeing the HiC map, and then you're seeing in green boxes the contigs, the sequences that are believed to be contiguous, and then in blue the scaffolds, sequences that are believed to form the chromosomes. But immediately, you probably, even if you don't know what it is you're seeing, you probably notice that there's this funny thing over here, this kind of like butterfly or bow tie-like pattern. And that's weird. You didn't really see any of that in the first talk. And what does this bow tie pattern sort of mean? And then there's another feature that you're seeing, which is you kind of saw domains before, but in this particular HiC map, what you're seeing is 2 blocks of the genome that form lots of contacts internally, but which never form contacts with one another. And if you just think about this from the standpoint of like physics, the physics of the genome, the kinds of simulations that I was showing you, having loci that are literally adjacent to one another in 1D, but never form contacts whatsoever, is thermodynamically impossible, right? This is not something that you see in any of the physical models that I've talked about, that anyone at this conference has talked about, that Jose is going to talk about. You're not going to see that sort of thing ever happen. So what we're seeing, and you will also not see these kinds of, weird bow tie patterns ever happen. These are things that just can't happen from the physics of 1D polymers. So what is it that we're actually looking at? And what we're actually looking at is mistakes in the assembly, problems in the underlying assembly. So let's start to analyze these problems. And my hope is just to sort of teach you how to analyze these problems. So when you look at a high C map, you're never fooled by these kinds of features. And in fact, you can exploit these features to assemble whatever genome you want, which, I mean, my hope is really by the end of this hour, if you decide that you are, desperate to, assemble the, Malaysian sun bear, this is something any of you guys could do for, not much more money than any other experiment might cost you. Probably the cost of a few textbooks. So anyway, so let's start to analyze these things with that in mind. So what's happening over here? So here we have two intervals of this genome that form lots of contacts internally, but although they seem to be adjacent in 1D, they never form contacts with one another. Well, that's really bizarre. And what you're actually seeing is a misjoin. So this is supposed to be contiguous according to whoever created this reference genome, but in fact it's not. It's a mistake. These were mistakenly joined or a misjoin. So the first thing that we want to do is we want to sort of, noticing that misjoin, we want to cut that contig into two pieces. And so here what we've done is cut that contig into two pieces. But of course, we still have problems once we've done that, right? We have this funny bow tie motif, this vertical and horizontal bow tie motifs. And what do these bow tie motifs signify? What they signify is that this contig, which was placed here, was not placed in the right order. In fact, this contig should actually be placed at that position. That's what we want to do. We want to take this contig, put it at that position, and once we do that, things look even better. But still, this is actually not a perfectly assembled genome. Do people see the remaining problems? Does anyone see the remaining problem? Yes. What's that? Sorry, like here? No, this is okay. That's actually fine. Don't worry about it. Yeah, so you're saying orientation. So what feature is cueing you to the fact that there's an orientation problem? Yeah, so that's a great point. So I'm showing you this sort of color reference, and what you're noticing is that there's this sort of weird discontinuity in the colors. So now this color reference is sort of a cheat because I'm using the fact that I know what the genome is supposed to be. But do you actually see what feature in the genome what feature in this map corresponds to that orientation problem? Yeah, so maybe I hope you guys see, there's actually this diagonal kind of bow tie pattern. And what that diagonal bow tie pattern tells you, just what was your name? What's that? Itena? Ikena. So what Ikena was pointing out is there's an orientation problem here. And this sort of diagonal bow tie is actually highlighting the orientation problem. What you're seeing is that the upstream bit of this contig likes to form contacts with the downstream flanking region, and vice versa. So what that tells us is that the contig, we put it in the right order, but we put it in the wrong orientation. What we really want to do now is, having noticed that, we want to flip it around. And once we flip it around, everything looks good, and we've actually successfully assembled two chromosomes of the human genome. And more critically, what we've learned is how to identify misjoins and ordering errors and orientation errors using high C maps. And the critical thing is, once you can reliably do all of those three things, then you can reliably assemble genomes with high C data. And I do want to point out that actually prior to our methods, there were no methods that actually allowed you to do all three of these things. And in particular, they couldn't correct misjoins. In particular, the results are pretty much catastrophic on real data if you can't correct mistruns. But finally, when we could do all three of these things very, very reliably, we had the problem solved. And you could really use high-C data for assembly. So one of the things that we did is we said, okay, let's, this was so visually intuitive, right, that Ikena was able, to quickly find these kinds of features, in the data himself, despite presumably only having seen this in the last few minutes. We said, this is so intuitive, we should create a system that allows people to take an extant genome and assemble it in this way, just on their own, just by eyeballing the data. We'll see how the sound works here. So we made a system for doing this. We find that like 8-bit video game music is really helpful when doing this, just to put the mind in a good, healthy place. So this is the Tetris theme song. So what we're looking at is actually the barrel clover genome. This is a genome that's published based on long read data and like dovetail data and bio-nano data, a whole bunch of different data types. It was published in late 2017, and it was a result of years of work. And what you're seeing is actually Olga Dudchenko, so my postdoc who really pioneered all these methods, and her work is just being sped up tenfold. So showing you 11 minutes of her work. You can see the clock in the upper right-hand corner sped up to about a minute. And what she's doing is first she gathered all of the contigs that came from the same chromosome, put them on the same chromosome, and now she's in the stage where she's taking each chromosome, putting all the contigs in the right order and the right orientation. And you'll see in a moment, she's now actually successfully assembled all of the chromosomes in about 11 minutes. This cost about $200 worth of data. So this is pretty good. I mean, they invested huge amounts into producing this genome. Surely at least, 3 orders of magnitude more money than the $200 we used to put the chromosomes together. So this is good. Not so good that we want to watch it twice. And what you can see is sort of on the left is where we started. Right, using this draft assembly of the barrel clover. And then on the right is a couple $100 worth of data, 11 minutes worth of work in this system we call Juicebox assembly tool. So Juicebox is our system of visualization. A set of visualization systems for high-C data. Juicebox assembly tools is the module in Juicebox that allows you to make all of these kinds of moves and does not yet actually have integrated Tetris music playing features, but we're working on that for 2.0. Anyway, sometimes you run into a genome that has like, instead of hundreds of pieces like the one I just showed you, it might have 100,000 pieces. In that case, no matter how loud your Tetris music is, we found that nobody really wants to do it in Juicebox assembly tools, and so for that reason we've created automated algorithms that do this. The automated algorithm is called 3D DNA, 3D de novo assembly. And what it does is misassembly correction and iterative scaffolding, and this additional step called overlap merging, which I don't have time to fully get into, but it's something that you also want to have. And what it does is it takes a high-C data set that looks like this, and it transforms it automatically, no parameter tuning, et cetera. You just run the code that's on the website, and it'll take this and transform it into that. And you can see that these are 3 healthy, nice chromosomes, and you can actually see the interactions between the telomeres. The telomeres tend to interact in this genome, and the centromeres tend to interact in this genome. So you can see all of those patterns. And I want to highlight that this is actually the problem that was on the front page of the New York Times, is the Aedes aegypti genome. And we actually had it on our hard disk because we've been working on it. And so we're actually able, within a few weeks of this appearing, to actually publicly share an end-to-end assembly of Aedes aegypti that actually solved the problem that was on the front page, so we felt good about that. I do want to highlight that this isn't total nonsense. So for instance, I'm comparing it here to linkage data. I don't want to get into details of linkage data other than that it's amazingly great. If you've generated high quality linkage data, it's amazingly great at telling you the relative ordering of loci in your genome. In general, when you compare the linkage data to a correctly assembled genome, what you should see is monotonic increase. And in fact, you see perfect monotonic increase here, except for like 1 or 2 or 3 outliers, which are well, complicated little story. So, the answer is that there's near perfect agreement between the genome that we assembled using the high-C data and the linkage data. I want to highlight that we did not use the linkage data in producing our Aedes aegypti genome. It's being used purely for validation. I also want to highlight that to make it clear, we took the best extant high-C based assembler from these proof of principle papers and used it to try to assemble the Aedes aegypti genome using the same exact draft genome and the same exact high-C data, and then compared it to the linkage data, and you can see the results here. What you're seeing is essentially that the results had nothing to do with reality. It's completely wrong. And it was just, these were proof of principle grade, but not actually capable of assembling a genome in the wild. So you can start to do analyses like this. I'm not going to spend too much time on this because it's not the central point of the talk, but all of a sudden, it becomes possible to start really expanding what you know about chromosome evolution in different clades. And so I just want to touch on this very briefly. So, we did Aedes aegypti. We said, look, while we're here, we should do Culex kinka-fasciatus, which is the principal vector of West Nile. So we upgraded Culex kinka-fasciatus, took a highly fragmented genome, produced chromosomes. Anopheles gambiae, which was the malaria mosquito, had already been assembled. So we had that, and we also compared it to Drosophila. Let's first focus on what's happening within the mosquitoes, because it's very striking. This is very different than what you see in mammals. So first, let me explain to you what you're looking at. So I've taken the Aedes aegypti genome and colored it in different sort of gradients. So that's what we've done here. And then we're looking at every single locus. This is sort of the legend, is actually. So it's the Aedes aegypti genome is sort of the legend for the whole plot. And then the same color gets plotted to whatever position it is in the target genome. So we look at all the little sequences of the Aedes aegypti genome. We say, where's the corresponding locus in Culex? Where's the corresponding locus in Anopheles? Where's the corresponding locus in Drosophila? And we map the color over there. One of the things that you see is actually Aedes is a much bigger genome. It's about 1.2 gigs. It's a much bigger genome than Culex or Anopheles. That's part of why people had Anopheles and they didn't have Aedes. Aside from that, the patterns of evolution that you observe are really striking. So what you can see is actually that within the chromosome arms, and I apologize, the projection is not perfectly sharp, so you missed a little bit of that. But within the chromosome arms, things are actually very scrambled. So this green is sort of scrambled up. The yellow is scrambled up. The blue is scrambled up. But the chromosome arms themselves remain intact across this whole evolutionary process. And in fact, just these three species alone diverged 150 million years ago. And yet, if you look at the contents of the blue arm in Anopheles gambiae, they're basically the same as the contents of the blue arm in Culex. They're basically the same as the contents of the blue arm in Aedes. Same is true for the purple arm and the brown arm, et cetera. The only events that really seem to have happened across these 150 million years is that the sort of yellow-brown arm, there was a single arm in Anopheles gambiae, sort of a mix of the yellow and brown arms in Aedes and in Culex. And there was also an arm exchange step between Culex and Aedes, where yellow ended up pairing off with red and blue ended up pairing off with green. So what's very, very striking is that things don't hop. The conserved elements basically are not hopping from one arm to the other. You're seeing scrambling within the arms, but really, really, really strong conservation of arm contents. This sort of thing had been, I do want to highlight, it had been hypothesized based on fish data before. We're able to see it very, 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 very clearly using this data type across all of these species. And I do want to highlight that actually the blue arm, if you notice in Drosophila, the blue arm is actually still there. So arm 2L in Drosophila basically has clearly corresponding arm, corresponds to 3R, 2P, and 2Q in Anopheles, Culex, and Aedes, respectively, reflecting the fact that arm 2L of Drosophila, for whatever reason, its contents have been largely preserved for 250 million years. And why and how that is a really interesting question. Okay, so we could assemble the Aedes aegypti genome. We have these 3D DNA automated assembler, and we have the juice box assembly tools, interactive assembler. So what are the different applications of this? So I want to highlight them. So one is we can, as I mentioned, upgrade genomes inexpensively. So this problem, you know, we spent about $1,000 on it, worth of data. In fact, we actually spent $0 worth of data, because the way we actually did this was we would just use the extra bits of sequencing runs that were full of other stuff and produce some data for this. So we generated more data than we needed to. But generally, these kinds of upgrades cost a couple $100. And this works pretty much regardless of the draft type. So here I'm showing you 7x high C data, so a couple $100 worth of high C data for a mammal, and combined with all kinds of, you know, short read and long read and phosmid, mate pair data, et cetera. And, you know, regardless of the data type, you end up with nice chromosome length scaffolds. So it's, yeah, so I mean, you get chromosome length scaffolds regardless of the data type, although the better data types will fill in a bunch of the gaps and thus your contig N50, the length of your contigs, to measure the length of your contigs, will be higher. We can also, when you have long reads, generate sort of platinum grade assemblies. And so actually the 80s Genome Working Group sort of nucleated in around the time of this Times article to try to make a much better 80s Egypti genome. And so they generated like every single data type from 80s Egypti. And so we worked with them to create an end-to-end assembly. So we're able to sort of upgrade the assembly from Dichenko et al. In this paper, Matthews, Dichenko, and Kingan et al. In Nature a year later. Now you can see, so I'm sort of mapping it the same way I was in that previous figure. You can see that basically the genome is the same, but a lot of there were about 25,000 gaps in the original genome that we upgraded. The number of gaps when we used sort of all these data types was down to 173. I do want to highlight that actually we only ended up using long read data plus the high C. Those are the only two data types that we actually used to make these assembly, And you can see we reduced the number of gaps to about 173. So all of a sudden you have very, very few gaps in these genomes. I do want to highlight that, you know, all these different data types like 10x read clouds and physical maps and optical maps and flow cytometry and SNP chips, all of this was generated. But we found that really the genome didn't get any better if you just took long reads in high C and then polished it a bit with Illumina DNA-seq, you got your pretty much optimal genome. And then we used all this stuff for validation, but it didn't change the genome at all. There was not a single case where the validation data contradicted the high C data and led in a way that led us to believe that the high C data had led us astray. So these things were used for validation, but didn't actually modify the resulting genome. And so what that highlights is if you have long reads and you throw in high C data, you basically have an optimal genome. And so, you know, this enabled us to really validate our methods. Finally, though, you know, long read costs have been declining greatly. So these days, you can probably produce long reads for any species for about $20,000. But it's still the case that short read data is much, much, much cheaper. And if we want to do something like $1,000 genome or a genome for a couple $100, you can't really do it with long reads. They're just not inexpensive enough yet. But you can actually do it using these methods. So I'll give you a simple example of a recipe for doing this. So Illumina, when they declared that they had a $1,000 genome, what they were saying was that for $1,000 you could generate 400 million 150 base pair paired end reads. So you're reading 150 bases from both ends of a piece of DNA. And then what you do is typically you align that and back to a reference genome, that's genome resequencing. So we said, well, why don't we allocate those 400 million reads a bit differently? We'll take 300 million and you do DNA sequencing in the standard way, and we'll save 100 million, do high C on them. And then what we'll do is we'll take W2-RAP, which is a contiger, to produce a draft assembly from the DNA-seq, and we'll use the high C data to order and orient those contigs, produce a candidate assembly, and then finally review that candidate assembly with juicebox assembly tools and produce a result. So what I'm showing you here is a dot plot from a human genome that we created for $1,000 worth of short read data. So the dot plot is showing you the relative position of loci in the human genome, current human genome reference, HG38, versus in our short read assembly. A perfect result is sort of like this one, lower left, the upper right, just, sort of a continuous straight line. Pretty much that's exactly what you see here, except you see some hiccups. And that's just because nearly all the cases, just because we're not estimating the size of the centromeres. So what this tells you is that for about $1,000, you can assemble any mammalian genome you want, or you can reassemble a human genome. I do want to highlight again that you could not do this with other assemblers that previously, sort of existed. So took like the Lachesis assembler, you could more or less figure out with this data type which bits were on which chromosome, but not more than that. And if you took the salsa assembler, it was designed to work with long reads. It doesn't really do anything at all with this data type. It's interesting to compare this to the reference genome that was produced for $4 billion in 2001. Obviously, sequencing has improved, by leaps and bounds since then. Nevertheless, it's interesting to note that for about $1,000, you get a genome whose dot plot actually looks a heck of a lot cleaner. So the ordering and orientation is actually much more accurate than in the original draft human genome, despite the fact that it's obviously considerably cheaper. I do want to highlight that right now, we don't actually place quite as much of the genome onto the chromosome length scaffolds. And we're slightly gappier than the reference human genome, but we think with software updates, this will rapidly be improved. So to put things in perspective, what are the kinds of things you can do with this? Well, after some of the early genomes, people started to produce, because it was so expensive to produce end-to-end chromosome length genomes, people started to produce very fragmentary genomes. There's about 1,000 fragmentary genomes on NCBI that are publicly available right now. The vast, vast majority of the genomes that are available are highly fragmentary. The cost of upgrading every single one of these genomes using Hi-C is about $250,000. So, if you think about, most of these genomes cost more than $250,000 to produce individually. And today we think we can upgrade them all for about $250,000. And of course, when you can start assembling a single genome for $1,000, then you can start to imagine assembling very large clades in this way. So for instance, doing all mammals using a purely short read assembly strategy, get them up to roughly HGP, roughly Human Genome Project standards, would cost about $5 million. So it becomes very, very tractable. So for that reason, we decided to create a consortium called the DNA Zoo. And we started this back in 2014 when we realized that all of this stuff would be possible. And we reached out to many, many different sites that had samples. Initially, we needed really, really high quality samples. So we wanted to get stuff, that was really as fresh and as high quality as possible. So we reached out to many, many zoos and conservation organizations and academic institutions. So for instance, SeaWorld has been a long time collaborator of ours, the Smithsonian National Zoo, the Columbus Zoo, one of the largest zoos in the United States, the Bronx Zoo, many, many different organizations have contributed. And the Houston Zoo actually has been a very, very close collaborators of ours. And we said, look, whenever you take blood, in a typical zoo, they're not going to, it's just completely unethical to ask them to, put an animal under anesthesia to allow you to do a blood draw. But if they're a good ethical zoo, then they're going to do physicals on every one of their mammals above 3 kilos every year or two. And when they do that, they do a blood test. And it is much more reasonable to ask them, when you do that blood test, if there's a little bit of blood left over, could you send it to us? Or if you could possibly take an extra half ml, maybe send that to us. And so we started doing that and started collecting samples from many, many, many species. And we started to get support from Illumina and from Edicogenome and IBM and NVIDIA and Opentrons, liquid handling robot company, Mellanox, to build, sequence these things and build computers to analyze these things. So now we're up to about, with the idea that, you could, that in a relatively cost-effective way, assemble pretty much whatever you wanted. So just to give you a sense of where we're at with this, I apologize, this slide is actually quite outdated. Anyway, about 57 collaborating zoos, about 700 plant samples, 2,400 vertebrate samples, 400 some insects. We cover about, when the slide was made, about 600 vertebrate species, including 234 mammals. I apologize, this slide is actually quite out of date. I somehow took the wrong one. The current number is actually more than double that. So we've got about 480 different mammalian species that we have samples of, which is about 10% of the clade. And we now have Hi-C maps generated in over 1,000 species and cultivars. Here's Arena, Omer, and Zane Kolarek, who are key experimentalists leading the experimental piece of this, guarding our -80 with all of our samples. So what are the things that we're doing? We're taking, whenever there's a fragmentary genome, we upgrade, and we have a sample from that species, we upgrade it to chromosome length. Again, it costs a couple $100 a pop. If there isn't a fragmentary genome, we just make one of our $1,000 genomes for this. And so we've done common raccoon, Virginia opossum, the wombat, Tamar wallaby, ground cuscus, eastern gray kangaroo, I want to highlight that in many cases, we're able to dramatically expand the number of chromosome length assemblies available in a particular clade. So for instance, there was only one marsupial available prior to our work on this, which is the gray short-tailed opossum. And just on this slide, there's a cuscus, kangaroo, wallaby, and opossum. So, and I think we've done already like 10 marsupials. Lion King is coming out this week. We did the meerkat for fans of Timon. Timon will be reassured. It is much better genome now. Jenny Tung and Tim Clutton-Brock and others contributed some data and samples for that. We've done the black-footed ferret. Here's a donkey. This works across clades, not just mammals. Here's the band-tailed pigeon. This is a genome that appeared in fragmentary form in Science in November 2017, and we were within a couple of months able to upgrade it to chromosome length. Here's another bird. This is the greater rhea. This is the first chromosome length snake assembly, the Burmese python. We took this highly fragmentary genome and put that together. You could use this for plants, so if people are concerned about ploidy and stuff, this works well for plants. This is the pigeon pea. This is the chickpea. Actually, this is a collaboration with Rajiv Varshni, a really terrific plant genomicist. One of the things that's really neat about this is it's typically been the case that you only have one reference for a species, and then you start to do resequencing. But together with Rajiv, we realized, hey, that's really need not be the case. So instead of remapping all of your other individuals or plant accessions onto your reference, why not just create many independent references that are independently scaffolded? So we're doing like a whole bunch of like 9 different chickpea accessions this way. So if you're a fan of the chickpea or of hummus or of Mediterranean food, then we call this the Future of Falafel project. We hope to improve your falafel experience. Here's some peanut genomes. These are the 2 ancestral parents of the peanut, 2 different species that actually hybridized to create the modern tetraploid peanut. So here you can see actually that ploidy is not an issue. This was just published in Nature Genetics by the International Peanut Genome Initiative. So it was a lot of fun working with them. This is the draft they gave us. This is what we sent back to them. We have to date shared over 80 chromosome length genome assemblies. I want to highlight, before we started sharing these, there were about 32 mammals that were available at chromosome length. I think we've roughly tripled that. We share genomes typically at least one a week. I think just yesterday we shared the black-crested cormorant for the cormorant fans out there. Let's see. And the data is actually all shared without restriction at dnazoo.org. So you can take the data, publish on it, analyze it, do whatever you want with it. We actually have deep high-C maps for each of these data types. So in addition, so you can actually do 3D genomic analyses and all sorts of other things. You can follow us on Twitter at the DNA Zoo. You'll get at least one genome in your tweet thread every week. All the methods are open source. So just go to dnazoo.org/methods. You can download like basically this 40-page cookbook, which, I mean, it may not get a Penn-Faulkner Award for writing, but it will tell you how to assemble a genome all by yourself, inexpensively for pretty much any species you like. Certainly a mammal will work with no problem. So, yes, you should read that. It's a terrific book that Olga put together. So in conclusion, the 3D DNA suite of tools allows you to use Hi-C to rapidly and inexpensively upgrade any draft genome to chromosome length, including inexpensive draft genomes generated from short-read DNA-seq. All this is open source. It works with contegs from Illumina or Oxford Nanopore, PacBio, or whatever you like. The total cost of upgrading all NCBI drafts is $250,000, so you can really get a lot of bang for the buck with this. And we've used it to assemble chromosomes for 80 plant and animal species. We can achieve very high assembly quality. We're pairing this with long reads, so like only 170 or so gaps left in the Zika mosquito Aedes aegypti. We could do de novo mammals with chromosome length scaffolds for about $1,000. It takes about a week. You could do all mammals this way for about $5 million. Here's roughly where the time and the money goes. You need a lot of computers to do this also. And again, we've had a lot of buy-in from different organizations and companies. But I want to highlight that the samples are coming from this very, very large network all around the world. Really, really grateful. For instance, like Kelsey Pletch, who pretty much single-handedly got us every single mammal in the Houston Zoo just by bugging people. If you know people who have freezers full of mammals or other interesting samples, or you have freezers full of mammals or other interesting samples, or you run a zoo in your backyard, like the gorilla zookeepers we discovered in Los Angeles, who I cannot tell you their names or their addresses, but they're like 2 blocks from my in-laws, and they have a whole zoo in their backyard in apparent direct violation of municipal code, but nevertheless with fascinating critters. Anyway, if you're those people, please let me know so I can bug you too. And again, all these ideas really were nucleated by the Center for Theoretical Biological Physics in our group, and really pioneered by a CDVP postdoc, Olga Dudchenko. So thank you guys very, very much for your time and attention through this heroic effort of listening to me talk not once but twice in a row. So I applaud you. Thank you guys so much. Okay, we can try to just keep the Q&A relatively brief, and then people can just ask me questions during lunch, if that works.
What time we start at lunch?
So, there's a start at
Okay.
Yes?
Neanderthal, do you know?
Yeah. That's a good question. So more broadly, the question is, how bad can the sample be and have this still work? And we are working on that. We are not yet at the point where we can reliably do high C on Neanderthal species. Or I should actually say, we are not yet at the point where we can unreliably do high C on Neanderthal species. It's always a disaster. But we're trying. We're hopeful-ish. We're a little hopeful. But the issue is the degradation of the geometric information. So there's some DNA left, but is that DNA anywhere close to its original geometry? Yes?
What about something extinct with more data?
Yeah. We've got pieces of mammoth in the freezer. And we're working on it, but I would say CF previous response. But yeah, that's obviously something that we'd like to do. A big problem is actually getting high quality mammoths. It's like that's a whole operation, getting high quality mammoth samples. Crummy mammoth you can probably get on eBay, but really good mammoth is worth its weight in, I don't know, a valuable substance. Yes?
Can you estimate gap size?
Yes, we can. No, we have not released automated code for doing it, but yes, it's totally doable. And it's important to do. We just haven't released code for it yet. Terrific. So maybe if there's other questions, we can do them over lunch so that I can liberate people to enjoy their lunch. Thank you guys so much for your time and attention. Thanks.
I noticed that grizzly bears seem to have many chromosomes. Is that true?
I don't know. We can check though right now on the DNA Zoo website.